My name is Stephan Holl. I'm the Group CEO of Conscio, and we are very happy to be able to continue our webinar series on biosimilars today, considering the evolving regulatory landscape, which we consider really as one of the most transformative moments in this industry. So I will just open up briefly for a couple of minutes to share some general insights, and then we'll later hand on over to Mina Grguri from Medicines for Europe, who will share some insights on the Streamlined biosimilar development landscape. And then later on to our colleague from Conscio, Eliška Kohelová, who will talk about immunogenicity testing on biosimilars. And of course, afterwards or during after the individual presentations, we'll share the stage for some questions that we'll answer through the chat and question and answer tool here on the webinar. So just to share a little bit of background, why is this important? We really believe that we stand at a historic inflection point. Over the next 5 years, we'll have about 69 biologic products in Europe and over 100 in the United States that will lose patent exclusivity. This, just to give some general perspective, represents an 8-fold expansion compared to where we were roughly around 10 years ago. So the speed of exclusivity losses and the dimension also of the sales of the reference products is absolutely unprecedented. And yet, and I think this was very well researched by our friends at IQVIA earlier today, there's only a limited fraction of those that actually have biosimilars under development. So 29% of molecules only currently have a biosimilar in development in Europe. Why is that? It's because of historically extremely high development costs. And this gap between opportunity and action is basically the challenge that the industry currently faces. And why should we all paying attention to this? This is because there is actually a lot of movement as we speak, which ultimately will improve patient access to very critical care. It will make it a lot faster as well. So for now over a decade since the first biosimilars came to market, biosimilar development has been constrained by generally a very cautious regulatory approach with mandatory comparative efficacy studies, switching studies, and clinical endpoint requirements that, while ensuring safety, also extended timelines and inflated costs. Now, in April this year, the EMA signaled a major departure from this paradigm with this draft reflection paper that we talked about already in our last webinar. And basically generally proposing a reduction in the reliance on comparative efficacy trials, and instead prioritizing advanced analytical characterization and pharmacokinetic data. The rationale is scientifically very compelling because modern analytical techniques have become so sophisticated that we can now establish structure-function relationships with unprecedented confidence, making large-scale efficacy trials redundant for well-understood molecules. And now, just recently, in October 2025, the FDA followed suit with its own draft guidance that mirrors and in some respects even extends on the EMA's approach. So the FDA is now also moving away from mandatory comparative efficacy studies, focusing instead on analytical and pharmacokinetic evidence. Most significantly, the FDA has also committed to eliminating those switching studies for products seeking this interchangeability status. It's a little bit specific to the US, but that has been a requirement that has long been criticized as adding unnecessary complexity and cost without a proportional scientific benefit. So this convergence also between the world's two most rigorous regulatory bodies is not coincidental. It reflects just a maturing understanding of biosimilar science and a commitment to a more efficient path to patient access, which we consider super important, to the industry and most importantly to the patients. And the first impact is already visible in of this general acceleration. So in the first half of 2025, the FDA approved 12 biosimilars across immunology, oncology, and endocrinology, and that already is a significant acceleration. In the EU in general, over 90 biosimilars have now achieved approval, with more than 80 marketed products actively competing in the healthcare system. And this acceleration will continue, with the market expected to double over the next 8 years to around $690 billion. Another part is that the approval is only a part of the story. So in the US, as of mid-25, approximately 73 biosimilars have received FDA approval, yet only 48 have launched. And also this pretty significant gap between approval and commercial entry reminds us that sometimes regulatory clearance, while essential, does not guarantee market success. So there's patent disputes, there's pricing negotiations, there's state-level substitution laws that remain a pretty big barrier to entry, especially in North America. The EMA's emphasis now on pre-submission engagement and the FDA's focus on sound scientific justification for waivers means that the successful developers will be those who invest strategically in characterization upfront, build robust pharmacokinetic programs, and engage regulators early and transparently. And this is, we see this discussion today and the insights we can share with our speakers as part of that, right? To get to a deeper understanding how this process is run most efficiently, most transparently, and with the biggest likelihood for success. So with that, I'm very excited to hand over to Mina Grguri, who's kind enough to share her perspective on this new streamlined path for biosimilar development. With that, over to you, Mina.
Thank you very much, Stephan, and hello, everyone. It is my great pleasure to be here with you today because this is a very pertinent topic for this moment in time for biosimilar industry and me representing Medicines for Europe, which is the trade association representing the off-patent medicines industry, generic, biosimilar, and VAM industry. So we are definitely very passionate about this topic and have been doing a lot of work now for years and years, actually, even though we are seeing kind of fruits of our labor only just recently. But this just shows that we need to be persistent and always keep science at the forefront. So, as I said, my name is Mina Grguri, and I'm the Biosimilar Policy and Science Officer here at Medicines for Europe. So, from in the introduction, we heard that this is really a multifaceted topic, but I would like to provide some perspectives from my area of work and expertise. So, as I said, streamlined biosimilar development is a regulatory evolution that is fully backed by science and founded in strong analytics. A short overview of just what I plan to cover in my presentation. So just give a bit of an overview where we stand and also where we started, and then what entails streamlined approach in the context of analytical similarity and comparability. What is actually the utility of the CES, and what are the opportunities that this can be unlocked? And lastly, where the global regulatory community stands on this matter. So we in Europe really are proud to say that biosimilar medicines are the story of a European pioneering success. And we are now almost 20 years after the first worldwide biosimilar medicine approval, which happened in EU in 2006, with the established pathway in 2004. Then some 10 years ago, or a bit more, we have witnessed the first monoclonal antibody approval in EU in 2013. And now we are really going global, and EU and US uptake accounts for over 90% of the global biosimilar medicines market. So it's important to have in mind that this success is really a multi-stakeholder collaboration approach, and this is important to keep in mind whenever we are looking forward into the future as to what more we can do to provide a better patient access to these important life-saving medicines. So just to see and take the temperature a bit of where we are, the current challenges that our industry is faced with, biosimilar developers. So it's important to note that in itself, development and manufacturing of biosimilar medicines is really a highly intricate, sophisticated process, and it demands significant expertise, substantial investment, and biotech level operational costs over really an extended period of time. So if we do a bit of an overview of what are the main challenges for a sustainable biosimilar model, we can see that there is several aspects to that. So we have right off the bat high development costs, which means high upfront investments. These are really dictated by the high reference product costs and the costs of conducting the comparability efficacy trial, the so-called phase 3, which according to some estimates, accounts for a third of overall development costs. Then high operating costs, because in itself, biosimilar development is biotech, and we are dealing here with a lot of injectable products. Next is that there is a high concentration of biosimilar assets, meaning that there is really a high focus on certain therapy areas and certain types of molecules, such as blockbusters, those that yield high sales, and particular therapy areas, as I said. Then the competition in the field is quite intense, which then paired with steep price erosion leads to a slower return on investment, which is also dictated by market dynamics and regulatory frameworks. Next, we have a high market volatility in the market with fast product hopping to newer clinical or therapeutic options, which means that we are often faced that the fact that by the time some biosimilar is on the market, the standard of care has evolved to a different type of recommendation. And lastly, that the existing regulatory requirements can definitely pose an absolute barrier to biosimilar development. So let's maybe focus on the latter and see why we are so excited for the potential of biosimilar streamlined development and the opportunity it brings with itself. So let's start it with understanding what it is. So when we talk about streamlining biosimilar development, we talk about development and regulatory approval of biosimilar medicines based on the analytical and clinical pharmacokinetic data. So this image of this pyramid that you see is basically what it would entail, the data package that the regulator would need to assess and conclude on the sameness of safety, efficacy, and quality of a biosimilar product. Why do we believe in this? Is because today's regulatory science suggests that a robust analytical and clinical PK data is indeed sufficient to assess biosimilarity in most cases. So no trade-off in quality, safety, and efficacy. An approved biosimilar by a regulatory body really always maintains these standards. So we are quite confident that this approach is ready to become the new default in biosimilar regulation. And the reason why we believe this is that because this approach is science-based, it is founded on 2 decades of experience with marketed biosimilar medicines in the EU, but something that we often forget because we always need to remember that biosimilar medicines are biological medicines, and the regulatory experts have now dealt for over 4 decades with the regulation of biotech process manufacturing changes. So this is really a lot of experience, a lot of expertise that really backs this excellent reputation that biosimilar medicines have today in Europe, but also globally. So in this approach, analytical comparability is really the main foundation of evaluation of biosimilar candidates. Coming back to this pyramid that we mentioned in the last slide, but maybe a bit more in detail. So we start with physicochemical analysis, whose main task is to confirm similarity and proper control of all relevant physicochemical attributes. It is the foundation of comparable efficacy, safety, and including immunogenicity. Next layer would be the functional analysis, which confirms similarity in all relevant functions, but also ensures that the molecule will be equally safe and effective in all clinical settings. And the last layer of our analytical comparability assessment is the clinical PK, which demonstrates equivalent drug distribution, confirms that the quality attributes relevant for PK are properly controlled, and where applicable, that other product-related differences have no pharmacokinetic impact, and it provides additional data to ensure comparable safety and immunogenicity. So if we were to go this way, we need to think about the prerequisites that need to be fulfilled in order to be able to base biosimilar development on clinical PK data and strong analytics alone. So first of all, this biological medicine that we are looking to develop a follow-on product of must be fully characterizable. So the critical quality attributes must be defined. There needs to be extensive and comprehensive structural, physicochemical, and functional characterization. So if we think about which medicines this would be, we can see that this above criteria is fulfilled for all recombinant biotherapeutic medicines that are currently produced or in development. So if we think about specifically which these are, would be monoclonal antibodies and fusion proteins, cytokines and growth factors, bispecific monoclonal antibodies, and antibody-drug conjugates. So if we think about any major differences that could arise in critical quality attributes, we have learned through this extensive experience mentioned beforehand that the clinical efficacy and safety studies cannot really address these major differences if we compare a proposed biosimilar and the reference product. What a clinical efficacy study, so the CES, can only do is confirm similarity of a proposed biosimilar that has already been established at the quality and PK level. But if we have any concerns that remain due to a low quality or otherwise insufficient comparative quality or PK data, these concerns cannot be resolved by a CES. To expand a bit more on this, this is because clinical studies are far less sensitive than analytical studies to detect differences. So there is a growing body of published evidence of regulatory expert thinking all across the globe, but what we would like to really focus on, because of its importance in the context of how many regulatory experts across the globe were involved, we would like to point to the IPRP workshop. So the IPRP is the International Pharmaceutical Regulators Expert platform that has been established for a few years now. And this workshop that is mentioned here has been conducted in September 2023. And you can find online the official report of the said workshop, which really goes more into detail. But overall, their thinking is, and their conclusion is that both regulators and industry experts who have been involved in this workshop broadly agreed that the comparative efficacy studies are not sensitive enough to detect anything but a very large analytical difference between a proposed biosimilar medicines and the reference product. So therefore, we can conclude that they are not likely to be additionally informative with respect to the small differences typically observed in analytical comparisons. So basically, by the time we have any unjustified difference detected, we are already at the late stage of biosimilar development. So this is a practical limitation. And therefore, we would really like to convey this message that relevant and unjustified differences in quality attributes cannot be addressed by any clinical studies, and they would require continued optimization of the manufacturing process. So if a clinical study, a CES, would signal any kind of unjustified difference, we would really need to go back into the manufacturing process and look what went wrong there, what went wrong in the analytical aspects, and what really needs to be refined there. We have also mentioned that there may be situations that may require a comparative efficacy study, and we find them very limiting. But if we were to hypothesize, they might be required for new indications for which the reference product is not licensed for. New route of administrations. For example, if a biosimilar manufacturer would develop a subcutaneous presentation while the reference product would only be able to be applied intravenously. So for intravitreal injected products, so in the realm of ophthalmology, where clinical PK is not meaningful, and where descriptive safety data and demonstration of non-immunogenicity could require additional clinical testing. And also for products which are extracted or purified from natural sources, we can maybe potentially consider additional CES, such as heparins, which are highly complex and heterogeneous. However, the sameness of the starting material sometimes enables abbreviated approval pathways, because in the US, low molecular weight heparins are approved as generic medicines, and in the EU, they are approved based on analytical and clinical PD data, so they don't require a PK nor a CES for an approval. So these are some hypothetical situations in which a CES might be still relevant. So this approach, why is it so important, and why it is very important that it is implemented, is because, as we can remember beforehand, as mentioned, the current regulatory requirements can consist an absolute barrier to biosimilar development in certain aspects, and thus limit the patient access subsequently. So what are the opportunities once streamlined biosimilar development is widespreadly implemented? So firstly, increase access of biological therapies to patients. Then we would get a higher feasibility of development for a broader range of biologics, including situations where there is an excessive originator reference comparator medicine acquisition cost. So in order to conduct this mandated CES, the cost of the originator reference product to which we would be comparing our biosimilar would be really making for a non-viable business case. And also where we have limited patient recruitment due to the fact that we are here dealing with a rare disease diagnosis, or where we have complex biologics in question. There is an opportunity, of course, to provide critical savings to healthcare systems by increasing the number of biosimilar medicines on the market. So we know that the competition can lead to more savings by providing more cost-effective therapeutic options. But this is not a standalone measure that could provide any kind of result. This would have to be done in synergy with appropriate market access and reimbursement policies. And of course, given that we are a global industry, this would have to be an international convergence effort, so that there is no duplication of work, whereas a company would have to develop biosimilar medicines for markets and regulatory regions where the requirements would be so different that still CES would have to be conducted. So this would possibly lead to opening of the new markets. But important caveat to all of this is that development may be less costly for some, but not for all biosimilar candidates. So we already mentioned the MABs, the fusion proteins, etc. So the cases in which streamlining would be applicable, but we are now going into the realm of if we look into what type of biologics are going to be losing exclusivity in the coming years, we're going into the realm of gene therapies. We are going into the ADCs, into the certain designations that would still require assessment in patients, such as in oncology, we would still have to conduct a clinical PK in patients. And lastly, biosimilar development is still biotech development with all of its complexities, so it cannot really be in any case assimilated to the generic medicine development and its costs and manufacturing and operational complexities. The regulatory community really has published a lot on this topic recently, also leaning on all of this experience with assessing biosimilar medicines and in conjunction with the advancements of the analytical methods throughout the years. So if we look at where we started in 2004 with the first pathway, we had really limited knowledge on what we could expect. So there was a cautious approach, but recent publications do confirm that confirmative efficacy studies do not offer a new or additional evidence of biosimilarity that is not already provided by this combination of comparative analytical assessment and clinical PK, so by streamlined approach. By EU regulators, you can find several publications that look back on the EMA experience with monoclonal antibodies and the learnings from those interactions with these types of developments, and the support is quite strong. Then we have publications by the US regulators, Japanese regulators as well on board. In the UK, they are now already having this implemented in their existing guideline, but also the industry has been publishing on the topic for a while now. We can maybe point you to these two publications from 2020 and 2025. So there has been really a growing body of evidence to support this claim. Now, where we are recently with the regulatory consensus in the global sphere, as mentioned in our opening, we really can witness in the last few months a lot of outspoken support for streamlining biosimilar development by the regulatory agencies that we kind of most look to when it comes to the acceptability and setting the standards and what regions across the globe rely on. So the EMA has conducted a workshop in September hosted by the Biosimilar Medicines Working Party, where we had multiple stakeholders involved, both the industry and different EMA working groups. And this really key discussion has yielded this workshop report that you can access on the EMA website, where there is support for this approach as the next step in the evolution of the biosimilar regulatory pathway. Now, what is awaited is the finalization of the reflection paper of the EMA, where this was discussed, and we were hoping that in Q2 2026 we will have this paper finalized and out in public, which would then hopefully lead to a guideline revision consultation opening. We have also seen in October that the US FDA has published a recommendation for the industry that really enables the developers to implement streamlined approach if they refer to the recommendations they have provided in their paper. So this can also be found on the FDA website. It is very important that also the International Council for Harmonization has, the ICH, started the guideline development process for determining a framework to determine the utility of a CES in biosimilar development. So this is really an important step when we look to global harmonization, knowing the importance that ICH has in the global sphere of the regulatory expertise. As mentioned, UK MHRA and WHO have already revised their guidelines to accept streamlining in 2021 and 2022. And we are aware that there are several other regions and agencies that are considering adoption of the streamlining approach in their guidelines via foreseen revision, those being Argentina, Canada, India, just to name a few. But naturally, all the regions are looking to how this can be implemented and with what recommendations. So to try to sum up this very extensive topic, but the streamlined biosimilar development is strongly backed by today's regulatory science, which suggests that robust analytical and clinical PK data is sufficient to assess biosimilarity in most cases. So there would be no trade-off in quality, safety, and efficacy in a tailored biosimilar data package. If we would like to have you take away one sentiment, is that in a streamlined development, we are keeping what matters and removing what is redundant. So next, that any relevant and unjustified differences in quality attributes cannot be addressed by clinical studies, and they do require a continued optimization of the manufacturing process. So going back and seeing what went wrong there. That this immense potential that this approach can unlock can only happen in synergy with appropriate policies via a multi-stakeholder approach, and that the global regulatory consensus community building is on the way. It's backed by numerous scientific publications, and some have already taken the step and implemented this thinking in an updated guideline. So thank you for your attention. I really try to be concise and on point, but this is an extensive topic with really a lot of facets. So looking forward to expanding on any aspect anybody else would be interested in. Many thanks.
Thank you very much, Mina. I don't think there's any immediate questions, so perhaps we'll wait for those till the end, and I will now hand over then to our colleague Eliška Kohelová, who is part of our Biologics department in Prague.
So good afternoon, everyone. Thank you for introduction, Stephan. So my name is Eliška, and today I will try to guide you through immunogenicity topic in the context of biosimilars. I will only briefly touch on biosimilars and some regulatory considerations, as Mina and Stephan has already covered some of those very well. My main focus will be on different aspects of immunogenicity in biosimilar development. We will try to answer what is immunogenicity, how can it affect efficacy and safety, and how are ADA and neutralizing antibodies tested. After brief introduction of immunogenicity, we will move on to the implementation of anti-drug antibodies testing in pharmacokinetic studies, and we will also consider some practical aspects of implementation of immunogenicity testing into pharmacokinetic studies. And our last topic will cover some analytical challenges related to ADA testing using one assay approach. So let's briefly align on what we mean by a biosimilar and why certain molecules have become market leaders. So I will start with FDA definition, which says that biosimilar is a biological product that is highly similar to approved reference product with no clinical meaningful differences in terms of safety, purity, and potency. EMA definition is very similar. Both agencies are emphasizing the same principle. It is high similarity plus no clinical relevant differences in how the product performs. In this context, there is one very common question, and it is why we are using term similar, not equivalent. The key reason is that large molecules are produced by recombinant technology in living cells, and during very controlled processes, there can happen some minor changes. For example, some changes in glycosylation pattern or some post-translational modification, and these changes can translate, but cannot, into some clinical differences. On the right side of the slide, you can see the top 5 biosimilars based on projected 2025 revenues. Ustekinumab, adalimumab, bevacizumab, trastuzumab represent major therapeutic areas such as immunology, gastroenterology, oncology, and autoimmune diseases. The projected 2025 revenues highlight that ustekinumab biosimilar are expected to reach around $1.5 billion ahead of adalimumab. It shows both patent expiries and strong clinical uptake of novel biosimilars. So on this slide, I try to sum up some regulatory consideration. My aim is not to go through every guideline in detail, but I will try to sum up some key message, how the FDA and EMA set the framework for biosimilars and immunogenicity assessment. So let's look in detail what is defined or how the guidelines are emitted by FDA and how the guidelines are emitted by EMA. FDA relies mostly on broad cross-cutting guidances. These guidances usually cover scientific principles of similarity, clinical pharmacology, and immunogenicity, but they usually don't issue product-specific or class-specific guidelines. In contrast, EMA uses core biosimilar framework supplemented by product-specific or product class-specific guidances. Good example is the guidance for insulin analogs. It could be said that FDA pathway is more flexible and general follows a totality of the evidence approach, while EMA pathway is more structured and class-driven. We can assess the impact of this strategy in the approval numbers. As of the end of May 2024, EMA has approved just over 100 biosimilars compared with approximately 50 biosimilars in the US. In this context, it is fair to mention that EMA started with approvals of biosimilars in 2006, whereas FDA started almost 10 years later. In last 5 years, the approval rates are relatively comparable across all agencies. What is also relatively interesting to mention is the situation in UK. You can see relatively high numbers for UK, 93. The numbers is the total number of approvals which were provided by EMA and UK agency. at 2021, there was a Brexit. That's the reason why this number represents the sum of both of EMA and MHRA approvals. So now I would like to revise how EMA and FDA expectations for biosimilars have changed and what that means for PK and immunogenicity study design. So as it was well covered by Mina, in the past large comparative efficacy trials were almost always required. Currently, these trials should be booked for very, very specific cases. For example, when structure-activity relationships are not fully understood, or when critical quality attributes aren't fully characterized, or when pharmacokinetics is not a reliable surrogate. When we assess current core evidence package, we can say that the basis is strong analytical similarity and well-designed pharmacokinetic similarity study together with comparative immunogenicity. It means that immunogenicity testing is not longer secondary, it's central. So regulators emphasizing a robust comparative immunogenicity and pharmacokinetic program, they expect really sensitive and validated methods for both anti-drug antibody testing and also neutralizing antibody testing. Appropriate timing and duration of sampling has to be set. And other titers has to be compared. So now I would like to describe some immunogenicity basics. So what is immunogenicity? Immunogenicity is ability of biologics to trigger an adaptive immune response, which is undesired. Immunogenicity is characterized by development of anti-drug antibodies, ADAs, and we know that anti-drug antibodies are able to bind to different parts of the molecule and may or may not have clinical consequences. We know that there are three main groups of factors influence these immunogenicity risks. First, patient-related factors, such as genetic background, concomitant illnesses, especially illnesses of kidney and liver. It can be also prior exposure to the drug or some pre-existing immunity, which is typical, for example, for IL-15-based therapeutics. Very important is also immune status. For example, when patients have some background immunomodulating therapy. The second category of such factors is represented by treatment-related factors. Dosing schedule, route of administration, frequency of administration, mechanism of action of the drug, and duration of the therapy. These are factors which can significantly affect development of anti-drug antibodies. And the last but not least, it is category of product-related factors. These factors are especially critical for biosimilars because you have influence over these factors. These factors include molecular structure, glycosylation pattern, some changes in the formulation, type of storage, downstream processing. Very important are also impurities, concomitants, contaminants, and solubility and stability. The key message is that immunogenicity is multifactorial. So when you design pharmacokinetic and ADA strategies, you should consider who you study, how you dose, and how the product is made. So in previous slides, we have defined what is immunogenicity and factors influencing immunogenicity. And now we will focus on why it matters in practice. What happens when patients develop anti-drug antibodies? So when patients develop anti-drug antibodies, there are basically three main concerns. The first concern is adverse reaction. The second is impact of ADAs on PK profile. And the last is neutralizing capacity of anti-drug antibodies. So let's start with safety. Antibodies can cause hypersensitivity reaction. They can cause also immune complex disease, which can be potentially very dangerous. They can inhibit a native protein or receptor. This process is typical, for example, for IL-15 therapeutics, erythropoietin, etc. To less severe adverse reaction belong some weak allergic reactions, but then we have also very severe adverse events like cytokine release syndrome. The second is impact on pharmacokinetic profile. So we know that development of anti-drug antibodies can cause aggregation of the drug. It can bring some steric changes, and this can lead to increase in drug clearance, thus decrease drug exposure and reduce clinical efficacy. The second thing, which can also lead to decrease in clinical efficacy, is neutralizing effect of anti-drug antibodies. Neutralizing antibodies can block target binding, so the drug stops working even if we still observe some detectable or measurable concentration of the drug. Both categories, neutralization as well as impact on PK profile, could lead to loss of efficacy of your drug. So now I would like to show how ADIs are analyzed and detected and why assay design really matters. So when we talk about ADA analysis, we are essentially talking about family of immunoassays with different formats and detection systems. So when I start with different formats, the most common formats is bridging and direct format. In the first setup, your labeled drug is used as coating and detection reagent. And in the middle, here we have anti-drug antibodies, which are represented in the assay by some surrogate positive control. The second very common format is direct format. In this format, we usually code the drug and detect using anti-human antibody or protein AG, depending on the drug which we want to detect or for which we are trying to develop the assay. So these setups can be run on various platforms such as mesoscale discovery platform with electrochemiluminescence detection, or you can also use classical ELISA setup with absorbance readout. You can use radio immunoassay or other technologies like GYROLAB, AlphaLISA, SPR, or BLI. As I have already mentioned, human ADAs are polyclonal and can recognize multiple epitopes on the therapeutic. So we typically use surrogate positive control antibody and report only semi-quantitative data, usually titers. It is important to note that key performance characteristics like sensitivity are relative values which are determined by the choice of surrogate positive controls. So it is really important to say that ADI results are in this context really assay dependent. Different formats and platforms can give very different ADA titers for the same drug, which makes cross-study comparison very difficult. A good example is adalimumab, which was used in treatment of rheumatoid arthritis. And when ADAs against adalimumab were measured via different setups, ADA in the range from 1 to 66% were observed depending on the assay and positive control used. So this witnesses how important it is to ensure head-to-head testing of the immunogenicity within clinical trial, as it is really complicated to compare your data with some historical data. So each ADA assay platform offers advantages and also limitation. Platform selection should be driven by the molecule, by expected ADA characteristics and study objectives, rather than some convenience and habit. When I start with ELISA, ELISA can be conducted in both setups, direct and bridging. ELISA still remains a widely used platform because it is very simple, cost-effective, it offers high throughput and high throughput. The weakness of this assay can be high background, relatively high chance of false positive results, and time to time this assay is not suitable for detection of ADAs against monoclonal antibodies. In case of bridging ELISA, this setup is slightly more specific and selective than direct ELISA. When we compare MSD platform with ELISA, MSD platform offers higher sensitivity and wider dynamic range, also weaker background signal. Both formats can be used, meaning direct and bridging. This method also offers high throughput. What is disadvantage of this approach is higher cost of the reagent, and you need to be equipped with specific equipment. Very old techniques is radioimmunoassay. It is sensitive and relatively inexpensive per test, but the use of radioactivity make it less attractive for long-term programs, let's say. And the last are label-free technologies like SPR and BLI, which are unique in detecting low affinity antibodies, and they also provide kinetic and isotype data. When I compare them with previous techniques, they are usually less sensitive and they don't belong to high throughput techniques. So in the context of ADAs, it is good to mention that both agencies recommend to conduct a tiered approach in assessment of anti-drug antibodies. The goal of this tiered approach is answer 3 simple questions in this logical sequence. So, is any ADA detectable? Is the ADA specific to the drug? How strong and persistent is the response? So when we start with the first tier, screening assay is designed to be highly sensitive. We typically accept a higher false positive rate, commonly 5%, because the priority of this tier is not to miss potential responders. So at this stage, we simply mark samples that show binding to the therapeutic and mark them as potentially positive. Samples that are potentially positive move to the second tier, confirmatory assay. Here is the main aim, specificity. Typically, we add access of the unlabeled drug to compete with ADA binding and confirm through ADA binding. If the signal drops as expected, we confirm that we have true ADA positivity. For the confirmed positive, we can determine the titer. The titer says what is the magnitude of ADA response. We perform usually serial dilution until the signal falls back to the cut point, which gives us an ADA titer and tell us how strong the response is. Depending on your development needs, we can add some further characterization if needed. For example, determination of neutralizing antibodies, description of domain specificity for multispecific modalities, or we can capture endogenous cross-reactivity if it is related to the mode of action of the drug. So now I would like to zoom in a specific subset of antidrug antibodies, neutralizing antibodies. So till now we have talked about binding ADAs in general, but neutralizing antibodies are the ones that actually block the biological activity of the drug by preventing its binding to the receptor. So basically we have two options how to analyze neutralizing antibodies. The first is via cell-based assay, and the second is non-cell-based assay approach using competitive ligand binding approaches. So the big advantage of cell-based assay is that they provide direct functional readout because they measure the biological effect of the drug in some relevant system in cell line. This approach is considered by regulators as the gold standard for neutralizing antibodies, but this approach has also its own weaknesses. As example, I can mention high variability of these assays, stronger matrix interference, lower sensitivity, lower free drug tolerance when compared to ligand binding assay. It is good to mention that it is relatively difficult to implement and validate this assay, so it takes some time. On the other hand, competitive ligand binding assays are at the other end of the spectrum. They don't give you a full functional readout, but they are typically easier to set up. They are more sensitive, precise, and they are more drug tolerant. They can be used when the biologics mode of action involves blocking of soluble target or blocking cellular receptor. So our central question in the context of biosimilar is, do the biosimilar and reference product differ in immune response, incident, or severity? You can see that immunogenicity is not just a regulatory requirement, but it's a matter of patient safety and therapeutic efficacy. From history, we know that preclinical evidence or some preclinical immunogenicity data are often not sufficient. We cannot fully rely on them in context of immunogenicity. In the context of biosimilars, there are two main clinical study design to consider. It is crossover design and parallel design. Crossover design is typically recommended for products with short half-life, with rapid PD response, and with low anticipated incidence of immunogenicity. In the context of crossover design, timing is almost everything. We have to carefully consider the onset and resolution of immunogenicity relative to the washout period. We know that anti-drug antibodies have relatively long half-life. It means that conventional washout periods may not be sufficient. We also know that common assays for detection of ADAs are not able to distinguish between ADAs generated to originator and to biosimilar. On the other hand, parallel design offers some advantages in context of immunogenicity assessment. Parallel design is basically more appropriate for products with really long half-life. When we plan repeat dosing, which can amplify immune responses. It is also suitable for diseases that show time-related changes associated with drug exposure. We know that many biological products have long half-life and elicit immunogenicity responses, and it makes parallel design preferable design in the context of immunogenicity assessment. So let's sum up the three main pillars that guide the design of a clinical immunogenicity assessment. It is a risk profile of the product, the study population, and the duration of follow-up sampling. So for the first box, we can apply a risk-based approach to the nature and severity of immunogenicity. So for high-risk products where antibodies could cause serious outcomes like anaphylaxis, neutralization of essential proteins, or loss of efficacy of some life-saving therapies, regulators usually expect a comprehensive and well-justified immunogenicity program, and sometimes also some post-marketing safety evaluation is recommended. On the other hand, for lower-risk products where immune responses are without severe clinical impact, some pre-marketing evaluation should be sufficient. So important topic is how to select the right population for this study. So the study population should be appropriate to detect meaningful differences in immunogenicity. The selected population and dosing regimen need clear scientific justification. It is important to remember that patients receiving some background immunosuppressive therapy are less likely to develop ADAs. So relying only of such patient population can underestimate true immunogenicity responses. The last thing which should be considered is the follow-up evaluation. In other words, how long is long enough to evaluate immunogenicity? Follow-up sampling should be long enough to capture development of anti-drug antibodies, their clinical consequences, and potential disappearance of transient responses. So these are factors related to the clinical study design, and there are also some analytical aspects. We should keep in mind that assay performance and sampling strategy are crucial. The immunoassays must be sensitive enough to detect clinically relevant anti-drug antibodies, even when some circulating free drug is present. This requires careful validation of free drug tolerance, and if your assay is not sufficiently, or if your assay doesn't tolerate sufficient concentration of the drug, you should consider some adjustment of sampling strategy. So this is my final slide, where I would like to go through how to compare ADA binding between the biosimilar and the reference product using one assay approach. This is a really critical step in ensuring the reliability and regulatory robustness of immunogenicity assessment. As it was mentioned previously, ADA assays are strongly dependent on selection of positive control. So to ensure head-to-head comparison of biosimilar and originator, you should apply this one assay approach strategy. In this approach, we should start with definition of critical reagent. Between critical reagent belongs our biosimilar drug, originator, labeled biosimilar, and polyclonal biosimilar antibody. This is our positive control. Next step is method development. The method should be established using labeled biosimilar as core reagent. We should perform one assay feasibility testing. It means we should test titration comparability and inhibitory comparability. During titration comparability, which is shown in the first graph, we usually demonstrate that the curves for low and high positive controls are comparable for biosimilar and originator. In other words, inhibition curves are generated using increasing concentration of the biosimilar and originator. The second comparability aspect is inhibitory comparability. Inhibitory comparability should test the assay's ability to detect ADAs across a relevant concentration range. In this case, we titrate positive control at defined concentration of the biosimilar and originator, and then again, we compare if these curves are similar. Once this one assay setup is established during method development, we can move to method validation. During validation, classical tests related to ADA validation should be conducted. This determination of assay cut point, sensitivity, matrix interference, selectivity, free drug tolerance, and stability, plus these 2 extra tests should be added to demonstrate that the assay works well for your biosimilar and also for originator. And that is
okay. Yeah, there is one now. I don't know who would like to take this from attendee. I work as an SME in a pharma-focused technology company, and coming from a pharmacology background, I'm curious, in your view, which stages of biosimilar development are the most demanding from a coordination standpoint, and which tend to be the most time-consuming or require the most repetitive work? Eliška, Mina, either of you? I think for us, as a service provider, we usually, we come in a little bit later into the picture, so it's a bit tough for us to answer, to be honest. But I think, Mina, any thoughts on that question?
Thanks. Again, I can maybe try and have a go at it, being that I'm more involved in the policy work itself and more on the regulatory side, less in the manufacturing. What this just kind of sparked is just maybe not exactly comment on this, but perhaps the fact that developers need understanding from the regulators that a lot of the development in biosimilar industry happens in parallel, that the stepwise approach that sometimes is maybe expected is not what is the reality. From a coordination standpoint, not really sure. I think, again, challenges can persist across different stages in the sense of, we mentioned how resource intensive is the existing requirement for a comparable efficacy trial. So I think that might be maybe one of the biggest challenges in the sense of obtaining the comparator product sufficiently, and then also finding the adequate patient to include in that trial, which is quite limiting. But then again, I think more and more focus should be focused on the analytics. So maybe there is where we kind of are sharpening the axe, let's say it like that. I hope this helped a bit, although it wasn't really exact, but I had to try.
No, I think that's very valuable. Thank you. Okay, I think, look, overall, thank you so much for the presentations. I think it really provided a great, overview of both the regulatory landscape and then the scientific challenges along the way that
Stephan,
will now.
sorry, we have one more question here. It came just now.
Okay. Yes, there's one more. So with phase 3 waiver coming into the picture from leading regulatory bodies, what will be your key advice to biosimilar developments? I think, I would like to give this question to Mina as well, but I think from our perspective, really, it's a little bit the reflection that I also started with, that we just have a belief that it's all about a very, very solid planning and a lot of transparency with the regulatory bodies, right? That really a lot of groundwork is being done, and a very strong case is built early on with the analytical data to then basically make the way to approval more predictable. And we believe that if that process is done consistently right, that then we will have this really tremendous new opportunity for many molecules that were previously not accessible or not developable, basically from an economic standpoint, and that's what we are quite excited to work on with our customers. But I will let Mina comment as well on this.
Thank you, Stephan. I mean, I can only agree. I think that what is important here is that since there is a lot of support from the industry side to go this route, we need to the developers need to show that they are really ready to provide equally safe, effective, and of high quality medicines in a tailored approach. So do not compromise on the analytics, even though utilize the the EMA scientific advice, any kind of pre-submission consultation that you can have with a regulatory body, because to discuss these concepts, to build on this practical evidence, because how change happens is that industry needs to really stand behind it and show the results, and that also helps build the confidence in the approach, because we need to see it in action. We need to see an approved tailored data package. So my advice would be, on one hand, as you said, strong analytics, strong foundations, and on another hand, do go forward with it and showcase what you are betting on, Yeah.
Perfect. I think with that, finally now, just somewhat in time anyway, thank you so much for joining us today. And again, I think one thing that we'll learn is also how this really all plays out in reality now in the future, and how we can basically go from this general guidance to a more evidence-based approach and more clear framework. And so there's a lot of things to discover. I think some of the email reflections will be finalized early in the year, and we certainly are very excited to embark on some of the first projects under this new approach with our customers in 2026. So until then, we wish you all a very nice and peaceful end of the year, a great Christmas time, and connect with us if you have any remaining questions or with the speakers. And other than that, we'll look forward to the next edition of our webinar series, and perhaps actually even also a physical meeting that we are planning on regarding biosimilars early next year. Thank you very much, and have a great rest of your day. Bye-bye.
Thank you.
Thank you.